All right, so in 8.4, we are talking about one of the two steps of the main job that DNA does. So far we've discussed kind of the history on how we discover DNA, the structure of DNA, and then we talked about how it replicates itself, which is important for cell division. But we know that DNA controls all of our traits, right? All of our inherited traits. And so how does that information stored in DNA become a physical trait like freckles or hitchhiker's thumb or eye color or whatever the trait is? So 8.4 and 8.5 are discussing the process of protein synthesis. And protein synthesis can be broken down into two steps: transcription and translation. And so the first step of transcription is how we convert the information stored on DNA into an RNA molecule. So RNA is another form of a nucleic acid, right? DNA is deoxyribonucleic acid stored in the nucleus. RNA is ribonucleic acid. And so RNA's job, actually there's several types of RNA, but one main job of RNA is to carry the information from DNA to RNA, and then RNA out to the cytoplasm. So the central dogma is kind of like the term central dogma. When we use that, we mean like the basic philosophy, the fundamental understanding of molecular biology is that information, genetic information, flows unidirectionally, one direction only, from DNA in the nucleus to RNA and then finally into proteins. That's how information is transferred from our cells. So the DNA stored in our nucleus gets copied into RNA, which will get converted into a protein. Okay. And so for this to happen, we have really three processes. The first one we've talked about, replication, and that is, making copies of DNA every time the cell replicates. That's important, right? Every time we make a new cell, we have to replicate our DNA. Going from the message of DNA into the message of RNA, or using DNA to create a molecule of RNA which carries information, is called transcription. So just like when you transcribe something, you write it down in the exact language that you're hearing, right? So that's what transcription is. It's the exact language. It's the same nucleic acid language that DNA is in, but it's a copy of it, right? When you graduate from high school and need a copy of your grades, we call that a transcript. Why? Because it's a copy of your grades. And so that's what transcription is. It's just copying the message of DNA into RNA. And then that last step from RNA to a protein is called translation. And so translation, when we use that word, we usually are talking about going from one language to another. And so that also makes sense here because we're going from the language of RNA or nucleic acid into the language of proteins, which is a bunch of amino acids linked together. So we're going from one molecule to a different molecule, but using the instructions in RNA to create that protein. So the original instructions come from DNA, right? DNA has the original instructions for how to make this protein. And that protein might, create brown eyes or whatever it is. Maybe it's the instructions for how to make melanin, which gives, your eyes the color they have. Well, how do we get that information from DNA and turn it into a protein? Well, we need RNA. RNA is that link. And so that's what transcription is. It's the copying of DNA into RNA. So RNA, although it's a nucleic acid, just like DNA is a nucleic acid, it differs from DNA in three big ways. First of all, RNA is called ribonucleic acid, which means that it has ribose as its sugar. DNA stands for deoxyribose. So deoxyribose is the sugar in DNA. So there's a difference in the sugar that makes it up. RNA also has four bases: adenine, cytosine, guanine, but instead of thymine, it has uracil. So when you're copying RNA from DNA, and remember we always use complementary base pairing. So let's say the code on DNA is AAA. Well, if we're going to complementary base pair, we would say, oh, well, then TTT should pair to that. But if we're making RNA, there is no thymine. So instead of 3 thymines attached to adenines, we would have 3 uracils, so UUU, because there is uracil in place of thymine. And also then RNA is a single-stranded structure. It's not double helix, it's not a two-sided, ladder. RNA is just a single strand. Okay, so there's our uracil instead of thymine. So transcription's job then is to copy the DNA into a strand of RNA like we said. So how does this happen? Well, there are genes on DNA, right? Genes are the information for a trait. So to get that information from that gene, we need an enzyme called RNA polymerase, right? It's the enzyme that's going to make my RNA polymer, or my molecule of RNA made out of many smaller subunits, many smaller nucleotides. So that's why we call it RNA polymerase. So transcription happens because of RNA polymerase. So RNA polymerase and a lot of other proteins, and it's a pretty complex process, but we're going to try to dial it down here a little bit. RNA polymerase and some proteins make what we call a transcription complex. It's like this machinery that's going to bind to the DNA, and it's going to read that DNA, and using that instruction, it unwinds that segment, that gene segment, and using the information on the DNA, it will complementary pair, as you can see what's going on here, right? If this must be my my template strand for DNA, I will RNA polymerase and the other proteins pair up RNA nucleotides. Now, this is a temporary bond, right? These are just hydrogen bonds, so these don't last. But RNA polymerase bonds these nucleotides together, creating the sugar phosphate backbone on my RNA molecule. And as this gene gets transcribed into, and we make our copy of messenger RNA, my DNA will wind right back up again, back into that helical shape. Once that RNA strand is made and transcribed from the gene, it disconnects, and now it's free to go out to the cytoplasm to get converted into a protein. And so in the process of transcription, there's actually 3 kinds of RNA that we're going to make, because we're going to need all of these RNAs for the next step of protein synthesis, which is translation. But messenger RNA, or mRNA, is what we are actually making during transcription. This, let's go back, right? This strand right here is actually mRNA, messenger RNA, because it's carrying the message from DNA that we're going to translate into a protein. The second type of transcription would be ribosomal RNA. Ribosomal RNA's job is to help make up a ribosome. And if we remember what ribosomes do, ribosomes are what make proteins. So we need ribosomes so that we can read that instruction, read that code from messenger RNA and turn it into a protein. But so it's kind of weird. Ribosomes are just a big combination complex of ribosomal RNA and other proteins that are going to actually make proteins. And our third type of RNA then is transfer RNA. tRNA, actually, I think of it as taxi RNA, because it's going to taxi in and bring in the amino acids to the ribosome so they can be assembled into that chain of amino acids that make a protein. And that transfer RNA, and we'll talk about how it does that, that transfer RNA actually reads the messenger RNA and makes sure that that amino acid belongs there. So transcription, and replication, very similar really, because we're unwinding the DNA, we're reading the DNA, and we're complementary base pairing. There are some differences though, clearly, right? Replication is going to make a copy of all of the DNA, right? When DNA replicates, every chromosome, every gene on those chromosomes gets replicated into a whole new set. Transcription is only copying a single gene, not every gene, just the gene for whatever the cell needs to make at that time. Replication also only makes a single copy, right? It's just going to make a single copy of DNA so that, the DNA, the original DNA can, or one of the copies can go to the one daughter cell and the other copy can go to the other daughter cell. But transcription, sometimes you need lots of these proteins made. Maybe you're trying to make insulin. Maybe you just ate a huge candy bar and your blood sugar is through the roof. So your body needs to make a lot of insulin. Well, one copy of that gene is going to make one molecule of insulin. That's not enough. So actually transcription makes many, many, many copies to get the job done of whatever kind of protein we're trying to make.